UK Wine Show number 864. The science of grapevine genetics, from clonal selection to CRISPR. Hi, my name's Chris Scott.
And I'm Jane Scott.
And together we founded 3050 and the UK Wine Show. On this week's show, I'm in Lincoln talking with Dr. Darrell Lizamore, Principal Research Scientist for Grapevine Improvement at New Zealand Winegrowers. We go right back to the basics on how grapevine genetics actually works, and from old-fashioned clonal selection through to CRISPR gene editing. I hope your week's been good. I have finished another round of feedback for our new website, and I was up in Birmingham for our daughter's graduation, so we're a little bit behind the time, but I think I'll get the show out in time. It was a great day. My daughter passed her Masters in Science based in physics, and Jane and I had a lovely time in Birmingham, and then we went over to Cosby to look at the Cosby yarn bomb, where people deck the town in crochet.
Yeah, well, the link there was that our youngest daughter is doing a bit of crochet. We got chatting to some parents who mentioned this Cosby yarn bomb, which was it was a bit of a left field thing, wasn't it, Chris? But we had a good day out. But mainly the graduation was absolutely superb. And we're both very proud of Isabel's achievements. And she's going on to do a PhD. She's going off to Bath University and she'll be doing a PhD.
Lovely, lovely. Okay, Jane, let's get straight into the show, though. No time to muck around other than to say happy birthday.
Oh, yes, it's my birthday today. So we're off to the theatre tonight, going to see The Devil Wears Prada. So I'm dragging Chris along and both daughters for an evening out at the theatre.
One of your favourite TV movies?
I absolutely love that movie, so I'm hoping the stage show will be just as good.
Alrighty, AJ, let's get straight into this week's Patreon multi-choice question. Every week we have some multi-choice questions for our patrons to try, and also 2 decks of flashcards. The multi-choice questions change every day, so you've got some activities to do as well as some flashcard decks to learn. This week's the decks were on, the level 3 was on Spain, and the level 4 was looking at the anatomy of the vine, shoots, buds, that type of stuff. But I actually chose a multi-choice question, and the question is:
which statement for Pinot Noir is not true? So the options are: Pinot Noir has thin skins, Chile's Casablanca region is known for producing Pinot Noir, Germany's Moselle is a key region for Pinot Noir, or South Africa's Walker Bay is known for its Pinot Noirs.
So here we go. How'd you get on that?
Well, I can see a few true statements there, Chris, so I've got to just ferret out the one that's false.
Okay, we'll give you the answer to that at the end of the show. But this week, there's a lot of doom and gloom in the news, and I was a little bit disappointed. You know, the people are going on about how wine sales are down 5% in 25, and Italy's got stacks of wine stored up. Sam Neill, the actor and owner of Central Otago's 2 paddocks, has died, and so I've decided to take some good news stories, but they all happen to be sciencey, which is fine because we've just been looking at physics and masters, and this week's show is a little bit geeky as well.
Okay, well, first up this week, a new study suggests that fermenting monastrell grapes under pressure, known as mild hyperbaric conditions, could enhance colour, aroma and structure without disrupting fermentation. While the findings are still awaiting peer review, the technique may offer winemakers an intriguing new tool. Researchers tested monastrell must under mild hyperbaric microoxygenation at 1.45 times the pressure of the atmosphere, equivalent to around 45% higher pressure than normal atmospheric conditions. Mild hyperbaric treatment places fermenting grape juice in a sealed, slightly pressurized chamber, allowing a controlled increase in dissolved oxygen without exposing the wine to excessive oxidation. According to the preprint, the process produced wines with deeper colour, higher phenolic content, and more pronounced fruity and floral aromas, while fermentation progressed normally and no sensory faults were detected. If confirmed through peer review and commercial scale trials, the approach would provide wineries with a non-thermal way to improve colour stability, mouthfeel and aromatic expression.
Well, we ferment wines under pressure all the time for sparkling wine production at higher pressures than this 1.45 atmospheres. But normally these are without skin contact. Now, I don't pretend to know the science in a great deal of detail, and what I can tease out and what I know is oxygen is needed often to stabilize anthocyan color molecules and to make them less reactive and stop the color fading in the wine quickly. Yeast also needs oxygen, so a little is good, but too much oxygen can make the wine fade and lose aromas. And from what I can understand from the research and my petty diving course I did years ago, because that's all about dissolved oxygen in your blood, the higher the pressure, the more oxygen is dissolved in the must, and the dissolved oxygen makes it easier to react. But it doesn't add more oxygen, it just makes it more reactable. And I think that's the idea behind this mild hyperbaric fermentation. Okay, Jane, what's the next story?
Well, next up, nitrogen levels in grape must could determine which yeasts dominate fermentation, with new research showing nutrient-rich conditions favour commercial strains, while lower nitrogen allows indigenous yeasts to survive and potentially add more distinctive flavours. Scientists have found that the amount of available nitrogen in grape must plays a crucial role in deciding whether commercial or indigenous Saccharomyces cerevisiae yeasts take control during fermentation. The findings suggest nitrogen management influences more than fermentation speed, potentially shaping a wine's aroma, flavour and regional character. The study compared commercial starter cultures with indigenous yeasts isolated from spontaneous fermentations using synthetic grape must containing different levels of usable nitrogen. Commercial strains consistently outperformed native yeasts in nitrogen-rich conditions, while indigenous strains remained competitive or coexisted when nitrogen was limited. Researchers also discovered that yeast strains differ in how quickly they absorb nitrogen and which amino acids they prefer. These differences help explain why nutrient levels alter the balance between strains. Although the work was carried out using synthetic must rather than must in commercial wineries, the findings could help winemakers fine-tune nitrogen additions to balance reliable fermentations with preserving distinctive local wine character.
Stuck fermentations are a real concern for winemakers. A stuck fermentation is when the wine starts fermenting and then before all the sugar is consumed, the fermentation stops. I personally think of yeast building up in a fermentation a bit like it's got momentum. That momentum helps towards the end of the fermentation when things get pretty inhospitable. As the alcohol rises during fermentation, it creates a more toxic environment for yeast. And most of the food that the yeast wants is gone. Add into it that wines have high acidity, and if the fermentation gets stuck, it may be quite difficult to manipulate to get going again. Also, yeasts that struggle often produce things like high levels of volatile acidity and unattractive sulfur flavors that you don't normally want. I remember talking to Siobhan Harnett. She was the viticulturist at Cloudy back in 2007 when I spoke to her. And she explained to me that adding nitrogen to a fermentation to make sure it doesn't get stuck is a bit like having McDonald's. It helps a lot in terms of food, but it's not a balanced diet. So playing with nitrogen and fermentation sounds super interesting, but it is a little bit like playing with fire. Nitrogen and yeast are really things that you need to get right in a fermentation. What's the last story, Jane?
Well, finally this week, South African researchers have developed Africa's first CRISPR-edited grapevine, marking a promising step towards vines that are better able to resist disease while coping with growing water shortages caused by climate change. Scientists at Stellenbosch University and South Africa's Agricultural Research Council used CRISPR-Cas9 gene editing technology to silence the VVDMR6.1 gene. Early research found the modified vines were less vulnerable to downy mildew, one of viticulture's most damaging diseases, while also showing improved water conservation traits. The work, highlighted by Nature Biotechnology, is significant because it targets two of the biggest challenges facing vineyards: disease pressure and drought. If confirmed through further trials, the technology could reduce reliance on fungicide sprays while helping vines perform better under increasingly dry conditions.
I think here the main consideration is consumer responses. We know a selection of genes in vines that give certain traits. Peewee wines is one approach of introducing them through sexual reproduction, and with speeding up things like genetic testing for the genes, it's about creating new varieties with enhanced genetics. CRISPR in this case is taking a gene, editing it so it no longer works. The grape is still the same variety, it's had a gene turned off. But CRISPR technology can add additional genes if scientists wanted. And this is similar to what Peewee guys are doing in terms of taking genes from other grapevines, not Vitis vinifera, and adding them to the Vitis vinifera grape varieties that we like. All these techniques are quite complicated, but this week's podcast, we lay out all the different ways that you can edit the genes or change the genes to create new varieties. But before we get into that, let's get a reminder of this week's Patreon flashcard question.
Okay, the question is, which statement for Pinot Noir is not true? Is it Pinot Noir has thin skins? Chile's Casablanca region is known for producing Pinot Noir. Germany's Moselle is a key region for Pinot Noir. Or South Africa's Walker Bay is known for its Pinot Noirs.
Okay, we'll give you the answer to that at the end of the show, but next up I've got the interview with Dr. Darrell Lizamore. He is the Principal Research Scientist for Grapevine Improvement at New Zealand Winegrowers, and where he leads up a team working on the genetics behind better grapevines. We explore all the known ways geneticists can alter the grapevine this week. Let's have a listen to what Dr. Darrell Lizamore has to say. Hi, my name's Chris Scott and I'm here with Dr. Darrell Lizamore. How are you?
That's also right. I'm well, thank you very much, Chris.
Excellent, excellent. We're here in Lincoln. It's probably, I think this must be either my first or second trip ever to Lincoln. I went to university in Christchurch, Canterbury, but doing engineering, but I never actually made it out to sunny Lincoln.
Well, welcome. Nice to have you.
What we're going to do today is we're going to talk a lot about genetics. Your role here is?
My role is Principal Research Scientist for Grapevine Improvement. So lead the Grapevine Improvement Program for New Zealand Winegrowers.
And that's really on the genetic side.
That's right, yeah.
And you have got a brand new toy.
We do. We've got a few new toys. Probably the one you're referring to is a sequencer that we've had for a little while in the laboratory now. We're effectively a fairly new team, and what we mean by grapevine improvement is improving the genetics of grapevine, selecting better plant material.
What we're going to talk about today is, I want to set the scene because people hear about different things about genetics, but there's different approaches to improving genetics. Some are adding genes, some are taking genes out, some are just doing all sorts of weird stuff. So I want to set the scene and understand what the options are for a geneticist to do things. And I don't want to get into the ethics of it too much, but if there's something that's clearly bad or considered bad by certain groups, then maybe we can highlight that a little bit. But so let's start off with what people have done in the past.
Right.
And that is plant breeding, isn't it?
Exactly, yeah. So grapevines are effectively the world's oldest domesticated fruit crop, which is quite exciting. There has been grapevine improvement done on grapevines before there was that done on almost all other plant species.
Even wheat?
Wheat, probably earlier. So in terms of fruit crops, grapevine is definitely the first, but people were probably collecting seed from seed propagated crops and like wheat and certain vegetables a little bit earlier.
Mm-hmm. Okay, and what plant breeding, what do you do there?
Yeah, so plant breeding is kind of a catch-all term for the artificial improvement of a plant, and typically what the scientific term would be hybridization. You're taking two different individuals and mixing their genetics and collecting a diverse panel of offspring and in the case of plants, planting them out and then selecting a subsequent generation from what looks good out of that collection.
So the plants are going through just natural sexual reproduction.
Exactly.
And you're getting, and in this case, you're getting a seed and you're planting it in the ground and you're going, is this a good plant?
That's right. So every single bunch of grapes, every seed in those grapes is genetically unique, just like the children of parents. And if you grow up individual plants from them, they're all genetically different.
So in the old days, people would just get seeds from grapes, put them in the ground and go, oh, I like this one. This makes a nicer grape.
That's right.
Than the old one. But then people started getting a little bit more clever because they'd say, oh, I like this particular grape. Let's call it Pinot Noir.
Yes.
And go, oh, this one's a little bit different.
Exactly.
And then that was called?
In the nursery, we call them bud sports, and the process would be clonal selection.
Clonal selection, yeah.
So there are multiple commercial clones of all of the major grape varieties. They all come from they can all be traced back to one individual seed, the same seed. So all Pinot Noir plants come from one Pinot Noir individual seed that was once selected hundreds and hundreds of years ago. And what happens is as a plant grows, to grow more branches and leaves, it has to replicate its cells, has to make new cells. And every time a cell divides, it has to copy all of the DNA. And in the case of grapevine, there's about 1 billion letters of code. And so those enzymes that do that copying of the DNA, they are very high fidelity, but they make some errors. So as the plant grows, it's making small, random changes to its genetics. Those can also happen when the DNA is damaged, like from UV light, and then repaired incorrectly. And those random genetic changes lead, in some cases, to slight trait differences. So Pinot Noir is a great example because Pinot Blanc is actually not genetically a different variety. Pinot Blanc is a mutant. It's a plant that has a small point genetic change, has broken its ability to produce anthocyanin. The color that produces the red skins in the berries, and as a result you get white berries. Otherwise it's basically the same as Pinot Noir.
Yeah, so you'd almost say Pinot Blanc and Pinot Gris are clones of Pinot Noir, but we sort of treat them differently, don't we?
You would, exactly. So from a geneticist perspective, they're clones, but for a winemaker, they're new varieties. Sometimes, it's an interesting one, sometimes when the spontaneous genetic change drifts so far apart, you almost have to label it a new variety for winemaking.
Yeah, you see that with Gewürztraminer and Traminer and...
Exactly. But certainly Gewürztraminer and Traminer, it's spicy Traminer. The change there is not a color one, but it's changed the biochemistry of that berry to the extent that the wine it makes is so different. From a winemaker's perspective, they treat them as a different variety.
Yeah, exactly. Okay, and that's clonal selection. And we're not really changing the genes, although the genes are being changed. It's just that it's happening over time in the vineyard, exactly. And it can take hundreds of years for these things to come through. And someone goes, oh, I like that.
That's right.
And then they start propagating that new plant material, exactly. And all of a sudden you've got a different clone of the same grape variety.
That's right.
And that's the way a lot of genetics, both plant breeding and clonal selection, has been done up till, 50 years ago?
That's right, yeah. That brand, that varietal identity of Pinot Noir or Chardonnay has been so powerful in the market that people have basically gone to clonal selection for their improvements. And they would typically do that by going to very old vineyards because they've had time to accumulate that random genetic diversity.
They used to call it massal selection or mass selection?
Yes, so mass selection means rather than taking an individual that you are cloning through propagation, you go to a vineyard and take a whole lot of cuttings. And so you're propagating a set of individuals of that.
Oh, okay, so it's slightly different. So you haven't got a homogeneous vineyard you're planting out.
That's right.
You've got some varietal difference in there.
Yeah, some clonal diversity that you're maintaining.
Okay, now we're going to get into modern technology and genetics. So the next one is induced mutation, because that's something you guys are doing here. So what's that?
That's right. So because mutations are a source of useful genetic diversity, and because they happen slowly and at random, people came up with the idea of let's understand why mutations occur and actually make them happen at a higher rate. And it's crude ways of doing that initially were like the Atoms for Peace program, where they decided let's find some useful ways to use atomic energy, and they planted a whole lot of plants around sources of radiation. The radiation damages DNA. Sometimes the host organism can't repair it in the right way, and so you get a lot of diversity. And there have been a few different So in addition to radiation, there's chemical approaches that you can use, but basically it's taking advantage of that resource of genetic diversity and usually stepping it up a notch so that you have a population where, rather than maybe in a vineyard 1 in every 1,000 or 100 plants have some useful genetics, it's happened 100 times more frequently, so every plant is visibly different.
I mean, I looked at it and they've got terrible names. Chemical mutagenic. Is that right?
Yeah.
That's where you have a chemical, and I've been watching too much Marvel comics, I think. Hulk, gamma rays.
Yeah, that's right.
Hulk. And what's the other one? Tissue culture stressing as well.
That's right. Yeah. So it basically anything that damages DNA or puts stress on the genome can cause these random changes to accumulate at a greater rate. And mutations have such a negative connotation because typically they make things worse. Generally, over time, evolution has selected for things that perform really well, and so most random changes are going to make something worse. And people's context of mutations is usually things like cancer. Something has broken the DNA, it hasn't been fixed properly, and you have some quite bad outcomes. But mutations are also the source material for evolution. Where sexual recombination just shuffles what's already there, the new diversity has to come from somewhere, and it comes from these mutations over time. So all the good traits also come from random mutations.
I imagine the failure rate is quite high, so you're doing it and you might have to do thousands and thousands and then go, oh, that one, it survived.
That's right.
And then, it's got a trait that you want.
Exactly. So because most of the diversity is going to be worse, you have to produce thousands. It's basically, if you think of any trait like height in humans, if you have a clonally propagated organism like grapevines, they're all genetically identical for that trait, so there's nothing to pick an improvement from. But if you produce random diversity, a key trait for us is timing of bud burst because of the susceptibility to spring frosts. If you have a bit of diversity, some of those plants are going to go through bud burst a little early, some a little later, but now you have a bit of a bell curve and you can go to the edges of that collection of plants and select plants that have the trait that you want, as long as you have some source of diversity to pick from.
And then we start getting into gene editing, where you're going in and you're changing things. The one that most people get a little bit upset with is transgenic modification.
Yeah.
And what's that one?
So transgenics, the trans means across. You're taking some DNA from something else and putting it in. And one of the most amazing things about genetics is that the DNA code is functionally the same. It's the same molecular basis in all organisms. So you can take something from a different organism, put it into another, and it will work. It'll be expressed. So that's transgenic. When something's come across, it couldn't have happened through sexual propagation, so it couldn't have happened in nature because those two organisms couldn't breed.
That's right. So there's things like where they make fish glow in the dark or fluoresce or something, isn't it?
Yes, and that's mostly been done as a proof of concept. If you're doing anything in genetics, if you're developing the method, you need something that validates that the method worked. So those fluorescent proteins came from jellyfish, and people thought, if I want to work out, can I put something into even my grapevines? Using a fluorescent protein is a good one, because if the grapevine starts to fluoresce under UV light, then it worked. And then you can swap it for something more useful later on.
And that whole transgenic stuff really took off with the development of CRISPR-Cas9, which is the sort of molecular scissors, isn't it? So you can control where you cut the DNA and certain things.
That's right. So CRISPR-Cas9 is adapting a system in bacteria that is for viral defense, which removes something that's, in the case of the original use case, it was removing a virus. But you can remove something that you would normally be there. The advantage of the CRISPR system is you can target it to a specific site. So they take the enzyme from bacteria, they also take a guide RNA, which tells it what to cut, and then you can put those two ingredients into any organism, and then the guide RNA will tell it which gene to damage. So if we use radiation or chemical mutagens, they just damage things at random. The advantage of CRISPR is you can basically damage something very precisely.
Yeah, so if you wanted to turn Cabernet Sauvignon white, you could target the bit that makes the colours and go set it up, and then you could make a white Cabernet Sauvignon. And that's not transgenic in that case, is it? What's that called? Where you're you're not adding new genes, you're just knocking out genes.
That's right. And there isn't a good set of terms here, and that's part of the problem, in that gene editing means we've changed what's there. You can have transgenic gene editing, where you've had to introduce some new genes to do that, and usually what they're doing is they're introducing the CRISPR enzyme from bacteria, or rather than introducing the code for the CRISPR enzyme, more recently people actually take the CRISPR protein itself with the guide RNA, introduce just those two components, which is hard because they're really big, but if you can get those into the cell, they can make the cut without introducing any foreign DNA. So you can make a specific cut in a specific location of the DNA without introducing a DNA. Technically, that's non-transgenic gene editing, and the really interesting thing about that is it just makes a break, which is something that happens all the time.
It's like a clone, isn't it? But it's exactly what you want.
That's right.
Is there any examples that you might want in vines to do that for?
Yeah, so there are some great examples. People initially, when breeding and transgenics were the main options for plant improvement, there was a lot of focus on resistance genes that have evolved in one organism to confer resistance to a pathogen. But now with gene editing, people have started studying susceptibility genes that have to be there in the host for the pathogen to manage an infection. And there are quite a lot of those. Those are being found as susceptibility genes for viruses and powdery mildews in grapevine, for example. And so CRISPR would be a way of knocking those out, removing a susceptibility gene as opposed to introducing a resistance gene.
Okay, and what's that called? Does it have a particular name? Just gene editing or?
Yeah, just gene editing and targeting a susceptibility gene.
But not all genes need to be switched on and off. There's an area known as epigenetics.
That's right.
Do you want to explain what that is? Because that's different. That's not really changing the gene, but it's changing the way the trays are.
Exactly. So every cell inherits the DNA of the cell that it replicated from. So it has all of the genes. But the question is, why do some cells become leaves and some cells become roots? And that's because only certain genes have to be expressed to make the tissue and perform the functions of that tissue. And so epigenetics is a layer on top of the DNA which causes some of the DNA to be expressed and some of it to be silenced so that it's not being expressed. Controls which genes are being expressed in a specific cell at any one time.
Yeah, and epigenetics is one of the reasons, you can tell me, I'm not sure if I'm right
Sure.
here, but if you plant a grapevine in a particular location and plant it in a completely different location, sometimes the vine will grow differently, and that's epigenetics?
Part of it, yes. There's of course an environmental effect. If the vine's been exposed to more sunny days, the fruit will ripen faster. But also the vine, just in being exposed to higher sun, different microorganisms in the environment, does actually start to change over time which genes are switched on and which are switched off. So a member of our team, Chen Liao, went and collected the exact same samples from the exact same clone of Sauvignon Blanc in different parts of the country, and it did methylation sequencing, which is looking at the epigenetics of those samples. And although they have the same genotype, the same DNA, their methylation is specific to the region that we picked them from.
If you move the plant to another region and plant it, will those genes then get turned back to what they were? So if you swap those two plants around, would they start looking like the plants from that region?
That's a great experiment. And we've never done that precise experiment. We've got a sort of proxy for that in that all of the material comes from a small number of nurseries. So we can say, what if materials collected by the nursery in one place, grafted, and then sent to multiple different places? And then we see it does, over time, adapt to the epigenetic pattern of the location that it's found, that it's then.
Okay. Is there an analogy with humans? Is there one that we could use?
Yeah, I'm trying to think of a good one. Most epigenetic changes are in response to stress. And so there's a great study, I'm hesitant to say where it was because I'm probably wrong, that looked at mothers whose food intake had been limited in the war. I think it might have been in the Korean War. And through that exposure to low amounts of food intake, their epigenetics had changed and it had been passed on to their offspring, and their offspring had sort of an adaptation that had been inherited from the stress response. The strongest changes to epigenetics generally are from environmental stress.
All right, and then once we've gotten to that, there's a thing which I've never really come across before called RNA-based spray-induced gene silencing.
That's right.
So that's where you're spraying something, is that right?
Yes. So DNA is the recipe book. It's everything that, the information of how to make all the enzymes, which are the molecular machines. For traits to be expressed, the regions of the DNA have to be transcribed into RNA. So the genes that are being expressed become transcribed as RNA, and then the RNA is translated into proteins. So if you can interrupt that process, you can, without changing the DNA, you can either remove the RNA that's being transcribed or change the RNA that's being transcribed. And that can be guided by RNA that's applied externally. So it's sort of a signaling molecule that can be hacked into in an organism to change the traits that are expressed without changing the DNA.
And by changing the RNA of the plant temporarily, you change a molecule and that can change some traits.
That's right. You might want to slow down ripening or trigger a stress response, for example.
And RNA can be used, we call it SIG, you call it SIGs, is that right?
Yeah, some people do. It's often referred to as, technically as RNA interference, it's using these double-stranded RNAs. RNA is usually single-stranded in the plant, but double-stranded RNA is most commonly encountered by the host as viruses. So they recognize this unusual type of molecule and they start to chop it up, and then they chop up anything that looks like that. So they remove a lot of the RNA that matches the double-stranded RNA that was introduced.
When I was doing some research on this, it seemed that molds and mildews were quite a good target for this. If you can make the double RNA look like a mold and mildew, you can actually use that to attack or kill the mold.
That's right. That's often where it's been targeted. If you could find some kind of pest or pathogen, molds and mildews, and silence a targeted gene that is critical to their survival and sort of silence that. The other approach that we've been looking at is silencing one of these susceptibility genes, so in the host. So rather than editing it, removing it, just temporarily silencing the susceptibility gene. And in a collaboration with the University of Milan, we have a PhD student from Milan here at the moment. She's been studying whether you can introduce these double-stranded RNAs targeting susceptibility genes, and it looks like you can get some resistance to powdery mildew that way.
What I understand, and you can correct me, is it washes off and it doesn't really stick, and it needs to be applied a lot, and so it's still quite embryonic.
That's right. It's very early stage, and the big problem with RNA is it degrades really quickly in the environment. It's ubiquitous. There's more RNA than there is DNA. Plants have one copy of the DNA and they just make lots and lots of RNA. So it's really abundant in the environment, but it degrades super quickly. So it's stabilizing it, getting it into the organism you're targeting. Those are the big challenges.
Okay, I think that's everything, is it? Have I missed something?
We briefly mentioned cisgenics.
Yes, yep.
Yeah, so cisgenics is the name given when you take a gene from something the host could have hybridized with, could have been bred with, and put it in directly. So for example, you might want to develop a disease-resistant Sauvignon Blanc that is resistant to powdery mildew. There are other species of grapevines that have resistance genetics in it, but if you cross them with Sauvignon Blanc, you end up with new seedlings. They are very genetically different, and that's where these new disease-resistant varieties or pewees come from. But you can take just the resistance gene out of that species and put it into Sauvignon Blanc the way you would do transgenics. It's not transgenics in that you haven't introduced something that couldn't happen through breeding, but it has been done artificially. So that becomes a bit of a gray area of how it's regulated. You've achieved something that you've only introduced DNA that the plant could get through breeding, but you have done it in the lab and you haven't shuffled the genetics like normal sexual recombination would.
Yeah. In New Zealand, which ones have you been looking at here?
Yeah. So our lab isn't a quarantine facility. So most of the new tools, we have a very strange way of classifying what GMOs are in our country. And they depend on the approach you take. And if that approach was being used pre-1998, then it's not GMO. And if it's new, then it is GMO. And so we have intentionally used this random mutagenesis approach because it introduces random diversity. It's been done for decades. It does mean you have to make thousands of plants, so that's what we've been doing. We have gone through the whole process for gene editing except for making the gene edits. We can't do that. We'd have to get licensed, and we'd have to do it in a lab that is basically regulated as if it was outside of New Zealand. So just bringing them out of a quarantine lab is treated the same way as if you were to bring in a new species across the border. But we think that could be a potentially valuable new tool, especially this non-transgenic gene editing approach, where you make a targeted change of the same type that we make through random mutagenesis, but exactly at the site that you want.
Well, I think next week we're going to look at your Sauvignon Blanc 2 project, where you're creating all these different mutations and seeing what's going on there.
Sounds good.
All right, cheers very much. Great. That ends the podcast. It's quite a technical one, I think. Well, a little bit technical, but hopefully you understand how things are changing and the opportunities. And the question really now is, what types of genetic manipulation is acceptable to the consumer? Clearly, using transgenetics may not be what people are keen on. However, is it appropriate to use CRISPR technology to knock out certain genes, like we saw in South Africa in the news article, as a way of creating new traits? We're going to explore some of the work that Darrell's been doing next week, looking at Sauvignon Blanc. Radio Jane, let's get the answer to this week's Patreon multi-choice question. And the question was, which statement for Pinot Noir is not true? Is it Pinot Noir has thin skins? Chile's Casablanca region is known for producing Pinot Noir. Germany's Mosel is a key region for Pinot Noir. And South Africa's Walker Bay is known for its Pinot Noirs. So is there any that you absolutely know are true?
Well, I know Pinot Noir has thin skins.
Okay, tick that off.
I know Chile is well known for Pinot Noir, as is South Africa. The one that's catching me out is Germany. I know there that Pinot Noir is known as Spätburgunder, and it is grown in Germany, but I don't think Moselle is a key region for Pinot Noir. So I'm going to plump for Germany.
Okay, well, Germany's Moselle region. Germany does make Spätburgunder or Pinot Noir, but it's in the warmer southern area of Baden. And the Mosel is really all about Riesling.
Riesling, and the birthday girl got it right.
Yay.
Yes. It's one of those that you do have to think through, though, don't you?
Yeah, I thought South Africa, Walker Bay, because we don't think of South Africa because of its latitude as being cool enough to create quality Pinot Noirs. But the Walker Bay is right by the coast. It gets the wind of the Cape Doctor. It gets the cooling Benguela current that comes up. It is one of the coolest parts in South Africa, and you can do it. And Casablanca, well known. It's by the coast. It gets a lot of morning fog, sea breezes, that type of thing. And it's very, very cool and does produce some good Pinot Noirs.
Brilliant. Well, thanks, Chris. And thank you for listening. And as always, if you did enjoy the episode, do subscribe to the show. And a big thank you to our patrons for supporting us. And if you'd like to join our community or find out more, head over to patreon.com/UK Wine Show.
Until next week, cheers and good drinking.
The UK Wine Show is a weekly podcast brought to you by 3050, the wine tasting company. The show is produced by Chris Scott and the news is read by Jane Scott. To listen to other shows, visit ukwineshow.co.uk. Thank you for listening.